Good morning, everyone. I will present the status report of my country, Argentina. My name is Pablo Tubaro. I'm the director of the Natural Sciences Museum of Argentina. This museum is located in Buenos Aires City. It's one of the largest museums of natural history in Latin America. I work in the evolutionary biology of birds. I am an ornithologist. And I also have been involved in DNA barcoding since 2004. First as a member of iBOL, then as a chair of the All Birds Barcoding Initiative, and then as a member of the Scientific Steering Committee of the iBOL. I'm happy to say that Argentina is one of the countries that contributes a lot to the barcode library. So far, we have produced 115,000 barcodes, corresponding to more than 17,000 bins. As you can see here, most of the bins are arthropods, mostly Lepidoptera, Hymenoptera, Coleoptera, and Diptera. But we also produce an interesting number of vertebrates species, mostly birds and marine and freshwater fishes. We have a network, a barcode network, since 2008. This network is composed by about 100 researchers working in 20 different research institutes of our National Research Council, the CONICET. The network was organized around a fund provided by CONICET, which is called the Argentine iBOL Fund. This money supports several barcode activities, including the organization of a workshop, training workshop for DNA barcoding every year at the museum. This workshop is done with the collaboration of BIO. The last one was last month in September. The iBOL Argentine Fund also supported a small project, more than 130 projects right now, with the aim to get new specimens for tissue for the barcode project. And the iBOL Argentine Fund also supported the creation of 5 barcode laboratories to extract DNA and amplify CO1 and send the amplicons to BIO for sequencing. This was necessary to avoid the restrictions existing in Argentina for the exporting and importing of specimens and tissue. We have several main barcoding projects in my country. 3 of them are the barcode of the birds of Argentina, the barcode of Lepidoptera of Argentina, and the Mangletrap program. All the 3 projects are for building the library of barcode sequences of Argentina and neighboring countries. The projects are based on the Natural Sciences Museum, and the leader of the project are the Dr. Darío Lismayer and I. And most of the collaborators are people from our staff, researchers, technicians, and postdocs. The first of this project, the barcode of the birds of Argentina, started in 2006. So far, we have produced about 80% of the 1,000 species of birds living in Argentina. We accumulate a large collection of tissue from birds from Argentina and also from Bolivia and Uruguay. We have been collecting in every part of the country and also in Bolivia and Uruguay. And our goal for the future is to continue to making collections, especially in Bolivia, which is 50% more biodiverse than Argentina. We have also good possibilities of doing more research and collecting expedition in Uruguay, and I would like to say we are looking for a partner in Chile to expand the project to that country too. The second project, the Barcode of the Lepidoptera, include not only butterfly but also moth and microleps. We have been collecting, especially in northeastern part of the country, and we also make some collection campaigns to northwestern Argentina and also in Bolivia. In our collection, we have so far about 1,200 species of butterflies, corresponding to about 90% of the butterfly species flying in the country. And we also get a large number of moth and Microlepidoptera, but that represent a lower percentage of the still unknown number of species flying in the country. Our plan for the future include increase the collection in northwestern, central, or southern part of Argentina, and also continue working with Bolivia and extend the collecting campaigns to Uruguay and Chile. And finally, the third project is the Mangletrap project. We have been operating 4 sampling stations in northern Argentina, one of the stations in the South Atlantic forest in the Misiones province, 2 sample sites in the Chaco ecoregion, in dry Chaco and wet Chaco, and the other sample station is in the Andean forest, which is called the Yungas. In addition, we also set 2 Mangletrap also in Buenos Aires City as part of the Big City Life project. So far, we have analyzed 2 stations, 1 year in the Misiones province and 1 year in the Chaco Mangletrap. We expect to analyze in the near future the sample from the Yungas. The sample from the Misiones province, I should say, this global record. This trap, just one trap, 1 year, produced more than 8,600 beans, different beans. The perspective for the future include an increase in the sampling effort with Mangletrap, trying to cover 50% of the ecoregion present in continental Argentina. That means to set a network of Mangletrap in about 7 different ecoregions of the country, mainly in the northern and central part where most of the biodiversity exists. And to conclude, a brief summary of our strengths, weakness, threats, and opportunities. Our strength is especially that we have the commitment to do DNA barcoding, to move forward with iBOL 2, and we have the human resources, the collection, and we know how to do the work. But our weakness right now is that we are in the middle of an economic crisis in Argentina, and this have an impact on our funding sources, and especially in the National Research Council. This is, of course, a threat for our work, especially if the economic crisis turns in a political crisis and goes deeper. But the main threat is not the economic, I should say, is probably, surprisingly, related with the CBD and the Nagoya protocol. Because in my country, as in other countries in Latin America, That means that the government tried to pass new legislation, which in general make more difficult our access to the biological samples and moving samples to make our job. We are fighting against this kind of limitation, but is, you know, the future will see if we succeed or not. Finally, regarding the opportunities, well, as I said before, we have the people, we have the good relationship with our neighboring countries, especially Bolivia and Uruguay, and we have the potential to scale up our operation in those countries too. And my museum is also set as an official repository for material collected in our national park systems, and also a repository for a large biodiversity project for the oceanic platform in South Atlantic Ocean and in the Antarctic Peninsula, which is called Blue Pampas. So we see those things are good opportunities for get a lot of material for the iBOL Phase 2 project. Thank you.